and co-released 21 wheat and 3 oat cultivars. Dr. Ibrahim has more than 375 publications, including 124 refereed journal articles, 40 extension articles, 13 technical reports and book chapters, 116 conference proceedings, 49 station and annual reports, and 33 popular press articles. Dr. Ibrahim is a fellow of the American Society of Agronomy, Crop Science Society of America, and Texas A&M Agri-Life. Dr. Ibrahim is also the chair of the advisory committee for Texas A&M Agri-Life Agri-Genomic Laboratory. Dr. Ibrahim's presentation title today, Updates on Hybrid Wheat Work in Texas. So we are pleased to have you, Dr. Ibrahim, and yeah, please go ahead.
Thank you so much, Dr. Muhammad and Dr. Ahmed for having me today to talk about hybrid wheat. And my relationship with Asyut is, you know, has been very long since I was a student at Colorado State University and a postdoc, you know, hosting Dr. Kamil and Dr. Amar Adri, and later Dr. Izzat Mahdi at Colorado State. And then here at Texas A&M, I was the major advisor of Muhammad Badri, and I hosted Dr. Rasha Mahdi in my program as well. And I look to a continued relationship with this reputed university. With that, I would like to begin my talk, but first I would like to give credit to the names that you can see here. The project leader is Dr. Benziger, and myself and Jackie Rad and Bojaraj Baznet from CIMMYT are co-PIs. And then everyone else is either a student or a research associate. So we trained Anil Adhikari as a student, Batul Sadeh, Jordana Tarlag, Brandon Gerish on this, and Nithya Subramanian as a postdoc. And then we have Dr. Shuyu Liu, who is also a faculty member at Texas A&M University. So this is not my work, this is everybody's work. It's a teamwork. And often when I talk about wheat, and I assume that you can hear me well, please let me know if you cannot hear me and I can actually try to raise my voice even higher. Diversity is a basis of crop improvement. And really, I'm lucky and privileged to be in Texas because it's a big state and we have a lot of variability in terms of soils and rainfall and temperatures, climates and disease pressures. We have very diverse germplasm in terms of conventional, and that's our focus. We focus on cultivar development. As you will see later, hybrid wheat is just a small portion of what we do. We have a big program on synthetic wheat. We have a couple of lines that we are ready to release based on that program. And then hybrid wheat is added to add diversity to the program. And we believe that the future has a lot of room for hybrid wheat, because if we want to meet the demand of 9.7 billion people, you know, every crop yield has to increase. And, you know, with the limitations and the global changing, we see hybrid wheat as the best tool to do that. We have diverse breeding methods. I usually use the bulk hybrid, but we also use double haploid, the double haploid method. And we have a very big team. You will see that here in a second. I also believe that innovation is what keeps you going, you know, enjoying your job, you know, and avoid repetitive research and breeding. So you need to add a few exciting things every now and then. In terms of germplasm, in terms of end products, because we breed for bread quality, but also flatbread quality and tortilla quality. We have a big program on that. And also, you know, breed for new traits, for adaptation, for yield, and for adding value. We are beginning also a program on BNI, which is a biological nitrification inhibition to increase the nitrogen use efficiency. I'll talk about that in a separate meeting, hopefully. So things are very exciting, and all of you know that breeding is both art and science. It's a numbers game. So the harder you work, the luckier you get. The more you test, the more likely you're going to have good cultivars to release. And so high throughput is a key word in our program. High throughput phenotyping, genotyping, high genotyping for marker, assisted selection and by crossing and breeding, but also image-based. You know, this is becoming a very big program. I would say we have the largest UAV program in the nation in terms of wheat. We have a very good team for data processing and analysis, and we have software that put that data at the fingertip of the breeder, because it's just basically make us more efficient. It enables us to get more gain from selection and so forth. This is, by the way, a picture taken by the drone of a field of a cultivar called TAM 114. This is one of the best cultivars in terms of in-use quality. Whereas around it, you see the trials. So the trials that actually led to this cultivar are shown in the same picture. And it just summarizes the breeding program of Texas A&M University. We have a very big team. This is a team at the Small Grains Workers meeting. We have that every year in August. And so the focus of what we do is cultivar development. Yes, conventional cultivars, not hybrids per se. We also have a lot of research. So we have the physiologists, the geneticists, entomologists, the end quality scientists, name it. You know, all of us work just as one team for, you know, good synergy and collaboration and to leverage resources. And hybrid wheat is a small part of what we do. So I just want you to know that we are not the experts per se in hybrid wheat. We're trying to learn a lot of things about it. Most of the hybrid wheat efforts here in Texas are based on the chemical sterilizing agent, basically Corvazer from Saturn Union, which was bought by Azure Genetics. But we are also working a little bit on the cytoplasmic system. Okay, so basically we think of hybrid wheat as a new solution to an old problem. You know, you have seen the case with corn. Since the inception of heterosis and programs focusing on hybrid vigor, the yield of corn has just been almost you know, parabolic in its movement and gain in terms of yield. And for wheat to compete with corn in the future, we have to increase its genetic yield potential through the adoption of hybrid wheat technology. And, you know, this is on the left-hand side figure. Wheat is the purple color, and we just don't want that line to continue, the solid line to continue. We want the dotted line to continue to. Because we have 9.7 billion people to feed by 2050, with a current yield, annual yield increase of wheat of less than 1%, we cannot make it. It has to be above 1.9%, or even some say 2.5% per year for us to achieve that goal. And actually, in the face of climate change, so which makes it more difficult. And this makes hybrid wheat just the logical alternative to do. And so that's why we put hand together with Dr. Benziger in Nebraska and Bojaraj in Simit to work on the tools, developing the tools for hybrid wheat. But we have a lot of challenges because wheat is self-pollinating crop, and you have to force it to behave as a cross-pollinating crop. Also, as you have seen in the last figure here, the major success of corn is not only because of heterosis and because of high yielding inbred lines, but because of the mass, you know, because of the number of people working on hybrid wheat. You cannot compare the number of people working on hybrid wheat compared to hybrid corn. Only very few companies and programs here and there. I just know of my program and Steve Benziger's program in Nebraska, that public programs that work on hybrid wheat. So with this number of workers, we cannot make it happen. So we need more workers. We need to share the tools and knowledge among programs. Just to be unselfish and to share that. So whatever we put into the program, in terms of CMS lines, R lines, we are ready to share with the community. Markers for selection, we are ready to share with the community. And I think this needs to be the trend in the public and the private sector. We still don't have that genetic diversity. And in case of very good diversity, then we have problem of nicking between the A lines and the R lines. And yield heterosis in wheat needs to be close to 20% for us to make this technology work. So we continue to work on it. And heterosis is just like any trait, you have to breed for it. We need to continue to evaluate the floral traits every year because, you know, whatever comes in the program needs to be evaluated for anti-extrusion and stigma gape and all of that. So you don't do it once and that's it. You have to continuously do it. Also, if you look at the conventional system, which is a CMS system, it takes a lot of time to incorporate the TMOVV cytoplasm in the germplasm and also develop the R lines that have the desirable amount of restorer genes. You have to have more than two or more restorer genes, as I'm going to explain here in the talk. And of course, the ultimate goal is to have an effective and cheap seed production system, because that's a limitation for the producer, as they have to buy the seed. This is a slide that was taken from Dr. Benziger, just to explain to you the difference between the conventional CMS system and the chemical system. Basically, you need the B line to maintain the sterility, to maintain the seed of the A line, right? Because the A line on their own, they cannot self-pollinate, so they cannot produce seed on their own. So you have to cross to the B line to do that. So to produce the A line seed. And then that A line, which is cytoplasmic male sterile because of a defect in the mitochondria or chloroplast genes, need to be crossed to a very divergent line, you know, developed in a separate pool, which is called the R line. That R line most of the time has a sterile cytoplasm, but it has to have at least one dominant gene for fertility restoration. Okay, so once you produce that hybrid that you see here at the bottom can actually self, because actually you produce the F1 and that is what is sold to the producers. So when they grow it, that is going to self and produce seed. Okay, so that's basically how the three-line system works. So we are developing this in collaboration with Nebraska, but we are doing it very slowly. And our focus is on the chemical system based on Courvoisier chemical hybridization agent from Assur breeding. So basically, you don't have to have an A line. You don't have to have the TMOVV female germplasm. You can just choose any triticum I7 line that you call a B line that has the desirable floral characteristics and the good combining ability. Apply the chemical agent at Zyrox 34 when the head is less than 2 centimeters in length, okay, and grow it in proximity of the male lines, okay, that can cross with the B line in this case to produce a hybrid seed. And this system is working very nice. We have a very big crossing block in Texas. Dr. Benziger has a very big crossing block in Nebraska. We target about 600 F1s every year to test between the two programs. So, and as I said, this is a funded program. It began with some seed money from Dr. Benziger. I had seed money from our agency here in Texas. It was called the Monocot Improvement Initiative. That enabled me to begin the program. And then we competed for NIFA IWeb funding. We got it. Dr. Benziger led that. And then we had a continuation from NIFA. to do so. So it includes Dr. Benziger, and then myself and Dr. Rudd and Bodja Raj as the co-PIs. Then we have key personnel from other programs. We even have a consultancy team that is helping us. So we are really grateful and lucky to have all of those people to participate in the success of the program. The objectives of the FAIRIS project include screening 2 weed programs for the floral traits, and then to create the seed in the crossing blocks and to begin testing the F1s, and also genotype those lines going into the heterotic pools, to try to develop algorithms to allocate lines into likely pools for future testing and validation, which we are doing actually right now in the continuation. And then continue to create a series of CMS tester lines and the maintainer lines and a series of R lines. I'll show you that in a minute. In the continuation, we propose to validate heterosis from the previously made and predicted wheat hybrids in replicated trials, continue male and female parent line evaluation for characteristics needed to develop experimental and commercial hybrids, and develop the heterotic groups or patterns and test multiple mating designs for wheat hybrids, and continue with our CMS line development, which is happening but very slowly. So this slide actually summarizes everything we do. Just here's at a glance what we have done. So we have basically screened almost all of the program for the male and female floral traits. We have been developing the R lines. We have been actually mapping those restorer genes, as you are going to see here in a minute. We tried to use the F2 as a proxy to the F1 because we have more F2 seed, right? So we can grow it in more locations and the replications and so forth. And usually in the program, we have about 1,000 to 2,000 F2s. Anyway, they're grown as big plots. In just the conventional breeding program, we can always actually harvest the whole plot and look at their yield and look at their heterosis. Of course, it's going to be 50% of what you would see in the F1, but it will give you an idea. And through a lot of experimentation for heterosis, we have a high parent heterosis of 118% with an average of 15.4. But commercial heterosis is more important, and we have a statewide highest 43.8% heterosis, but the average across the whole state of Texas was 11%. Okay, so you can find things that are lower than 11 and things that are higher than 11, but when you average all of them, that is 11%. So let me elaborate here a little bit on those projects. So as I said, we screened the whole program for the male and female traits. In the female, we look at the stigma exertion, how the stigma shows through the blooms, the stigma feathers duration, because the stigma actually remains fertile and receptive for a long time. We had been measuring that. And then the gloom separation, which we call Gape, is another trait, because without Gape, the female pods are not going to be ready for the pollen grains from the male parents. Then we did a lot of work on anther extrusion. We have what is called anther extrusion per se, where we actually count the number of anthers. And then an anther score, where we visually assign a quick score. We found a very high correlation between the two. And then we went into the trouble actually of looking at anther dimensions, like the width and the length of the anther and the weights of the anthers. But I don't want to bother you with all of that data. I'm just going to focus on anther extrusion for the males and Gape and stigma exertion for the female. And of course, you have to measure the flowering time, because you need to make sure that the parents nick in the crossing blocks. Now, there are issues with vernalization and photoperiod responses, intrinsic earliness per se, and their interaction, and that sometimes can mess things around. So you need to be aware of any photoperiod effect on the earliness of your lines. You also need to look at plant height, because we like to have males that are taller in height than the females, just for the purpose of shedding the pollen grains, and you don't want the females to be later than the males for the same matter. So measuring flowering time and plant height is very significant. Just to show you what I mean by stigma exertions, of course, Gape means that the glooms start to, the lemma and pellia start to move away from each other to expose the stigma, and then the stigma exertion can be seen here in this figure from 1 to 5. The ease with which you can see that stigma coming out of the floret is what I mean by that is stigma exertion. So we found that we have very good variability for the male and female traits in the Texas germplasm, and Dr. Benziger has done the same thing through students in Nebraska. We have good repeatability for the days to anthesis. Plant height was moderately repeatable, and I think it has something to do with the sample size. Anther score and anther extrusion were really highly correlated, and they were highly repeatable. But it seems like our ability to measure the female floral traits is still not very refined. We are working on refining that because we have been struggling with it. Repeatability, of course, sets the upper limit for heritability, as you know. So you expect the repeatability values to be higher. Repeatability is also, you do it only for one location, right? But heritability is across locations. And so the repeatability numbers match the heritability numbers, meaning that we have very good heritability for the male traits, but not for the female traits. And as I said, we are working on improving that. Well, our evaluation of the female trait actually involved the application of the chemical at Zadux 34 because you want the anthers to be inactive for you to evaluate the female traits. Okay, so this is a summary of the floral characteristics. We have good variability for both. The significant and positive correlation between anther extrusion and score shows that we can just visually, quickly, without counting the anthers, we can actually determine the score and use it. High heritability indicate that the male trait could be improved by selection and breeding even across environments, whereas our methods for assessing the female trait need to be improved and standardized. We're still working on that. Okay, estimation of how did we estimate heterosis? As I said, commercial heterosis is going to be more telling of hybrid vigor than mid-parent and high-parent heterosis, because actually sometimes we get very high heterosis between two parents that are not necessarily the highest yielding. Like one of them is very high yielding, one of them is low yielding, and yet they give us very good heterosis. So I would not use mid-parent and high-parent heterosis to compare hybrid. I would use commercial heterosis, which is comparing the F1 yield, in this case, to the best commercial hybrid available out there. So I have some data to share with you. We began this long time ago with the work of Brandon Garish, who was a master student, and he did it in greenhouses and cross chambers under spaced planting conditions. And we were able to get a hybrid heterosis of very close to 200%. Mid-parent heterosis of close to 90% from a cross of TAM 113 by 32K. 32K is an old line, has very good disease resistance, but not very high yielding. So sometimes those, poor yielding lines cross to a high yielding parent might give you high heterosis. So it's very unknown. And then we started to do that under the field conditions in collaboration with Nebraska. We have done that on the F2s and the F1s. So this is a summary of basically This slide summarizes it all. Across the 3 locations in Texas, we have a high mid-parent heterosis of 118%. And when you average across the 3 locations, it was the highest was 18.6. High parent heterosis was 103%. And when you cross across the 3 locations, it is 15.4%. Commercial heterosis was as high as 43.8%, and across the state it was 11%. Realize that Town 111 is the most planted wheat in Texas, so it's still a very popular cultivar. Okay, so we then went to using the F2s as proxy for the F1, because remember, actually we did that first in the pipeline, the breeding pipeline, just looking at the breeding pipeline F2s in the field. And when we saw the promising results, we decided to do this as an experiment across 6 locations in Texas and Nebraska to test the possibility of using the F2 as a proxy for the F1 and even to rank the F1 and to see if it can actually predict heterosis well as the F1 does. So we had to analyze the location separate because we wanted the LS means, which are the blobs in this case, and then we use the blobs in this GG by plot. This is basically a which, when, where view. As you can see, those are called the vertex genotypes, 14, 38, 35, and 18. Okay. But then you have what is called the environmental vectors, those red lines that emerge from the origin. The angle between any 2 lines will approximate their association. So a narrow angle will show high correlation. A right angle will show lack of correlation, and obtuse angle will show negative correlation. So it seems like McGregor and Prosper, because both of those are in the black lands of Texas, they tend to behave like each other, whereas the Nebraska lines and to some extent the bushland location, and bushland is in the high plains of Texas. It has temperate climate. It has a Midwestern climate, basically. We have snow in the winter, very cool nights, and so forth. So to some extent, it tends to cluster very close to the Nebraska locations. So this shows the locations here for the F2 test. And we had very good correlation between the F1 and the F2, but they tend to be more specific to the location. So if you try to, for example, correlate the F2 at McGregor with the F1 at Lincoln, you don't see that very good association, but you see it within location. So we figured out that for local adaptation, the F1 can be used as proxies to the F2s very nicely. Okay, but if you want to use that technology for a hybrid that is broadly adapted, then the F2s might not give you good readings for heterosis. That's one point. So the summary for this part of the research is that the high correlation between the F2s and the F1 is there, but it is environmentally specific. Moderate rank correlation. So the F1s can be ranked using the F2s, and some inferences about mid-parent heterosis can be done, but they are very specific to the climate where these evaluations are being done. Okay. So this is the last part of the talk, and so far I think I use about 29 minutes, so I still have some couple of minutes. I'll be able to finish in time. So the fertility restoration study was actually planned because we needed the markers to develop better R lines. Okay, and so we have this line from Cargill, which actually restores really well. So we wanted to map those genes and tag them, and then later develop SNP markers around them, because we believe that development of those KASP markers can facilitate the stacking of those restored genes. Remember that one gene will not restore fertility fully. So you need at least 2 genes. In some cases, you need 3. Okay, so this cross is really a very interesting cross, and Dr. Benziger will call this the overlooked cross, because you only make it when you want to study the inhibition to restoration, or you want to map restoration per se. Here, the R line is going to be used as a female, right? So it has dominant nuclear genes, but it has a sterile cytoplasm. Okay, whereas the B line is just a triticum isostigmum line with it can has a cytoplasmic sterile genes, but it has to have the fertility genes in the, actually no, in this case it's a fertile germplasm, fertile cytoplasm. Okay, and it can has recessive fertility genes. So when you cross the two and then you develop recombinant inbred lines, those lines are going to vary a lot. They can, all of them can have the recessive cytoplasmic genes, right? But they will vary in the fertility restoration. Some of them are going to be homozygote dominant, some of them are going to be homozygote recessive, and some of them are going to be heterozygote. So you have different levels of fertility restoration. And once you cross that to a same-S line, an A line in that case, that will show you and actually will enable you to determine the number of genes and map the trait very well and understand any inhibition to restoration. So this is how we did it. Basically, cross an R line as a female to a B line as a male. And the work was done by Anil Adhikari, who was a student under me in Texas. Now he's working for Monsanto. So we did this in the greenhouse as well as the field. So the real per se evaluation, but we have also done test cross in the field. Okay, and based on that, we were able to map 3 genes or 3 quantitative trait loci. The first one was on the 1B chromosome, and that explained 35 to 45% of the phenotypic variability. That's an R square of 35% to 45%. And this is most likely, or for sure, RF3. This gene has been mapped before. And then we had another minor QTL on the 6B, which explained 3.4% to 7.1% of the phenotypic variability. Most likely, this is RF4. And then another QTL on 5A, which explained 6.4 to 11.4% of the phenotypic variability. Even though this has higher variability, it did not help as did 6B. So we found that if you put 1B and 6B on the same background, you fully restore sterility. You don't even need 5A. Okay, this shows the physical location of that QTL on 1B. And actually, we went ahead here and under Dr. Subramanian's leadership in the agri-genomics lab, for which actually I'm the co-director and the head of the advisory committee, we developed KASP markers for RF3 and RF4 that we can now use in our selection and development of those R lines. So to summarize this part of the study, the fertility restoration genes in Cargill 115 is at 1B, which is RF3. Minor effect loci present at 6B, which is 4A, RF4 and 5A. And Cargill 115 can restore fertility fully because of the RF3 and RF4 combination. RF3 by itself will not restore the fertility fully. RF4 by itself will not restore fertility fully. You have to have the 2. And as I said, we developed KASP markers to be able to precisely breed and develop our lines. And then, genes from this study can be helpful and can be shared with other programs. Well, the take-home message from this very basic presentation is the following. We believe that hybrid wheat research is taking off. We will not afford to let it actually lag. Learning from the stories of the past, we cannot let that happen. We have good variability for female and male traits. Our F1 heterosis estimates are really promising. We know that we can use, under specific adaptation conditions, we can use the F2 as a proxy for the F1. Major genes for restoration have been mapped, and we have developed KASP markers around them. Good progress is being now made for the CMS system, which is cheaper, but takes longer time. And we believe that collaboration and continued funding is very key to continued success. With that, I would like to acknowledge the USDA, NIFA, and the IWEB program. Texas Wheat produces both Texas A&M AgriLife Research, CIMMYT, Azure Plant Breeding, University of Nebraska-Lincoln, the Nebraska Wheat, Monsanto Bayer Crop Sciences, Monsanto Species Bolog, which actually covered the scholarship of Anila Dikari. And with that, I would be glad to answer any questions. Thank you.
Thank you very much, Dr. Amir, and for the great and informative presentation. Personally, I got a lot of information, and thank you very much. Kindly, I'm asking everyone, if you have any question, please raise your hand, and Dr. Amir is more than happy to answer it. So, any questions? Or
we can also wait until Steve gives his talk. And we can open it to questions that way after.
Okay, yeah, that's a good idea.
So I'm going to stop sharing the screen so he can share.
Yeah. So Dr. Steve can share it, his presentation now.
I think I unshared.
Okay. So Dr. Steve.
First of all, can you hear me?
Yeah.
Yes.
Okay, so now, and you can see my screen?
Just a second, yeah. So give me like a 1 second, Dr. Steve, just introduce yourself. I will do it, so yeah. So now we have our second great speaker. He's actually a well-known worldwide scientist, Dr. Peter Stephen Winziger. He's a professor emeritus in the Department of Agronomy and Horticulture, University of